Hi, James has come all the way from Oxford. Actually, he's always quite far. 2 trains?
Yeah, 2 trains.
Yeah, 2 trains. To come and talk to us about human genetics and child infection. He's just recently got a welcome CDA, started a couple of months ago. He's a paediatric ID physician by training. I know he's our local paediatric ID physician. And has done a lot of work, essentially, both in the UK and in lower income settings, using homogenetics as a way to sort of help understand the cause and consequences of infection. So you can talk a bit about your previous work and a bit about your CDA?
Yeah, I guess I'm sort of using my previous work to sort of tell you what my sort of worldview is and how that's going to inform what I'm going to try and do for my CDA. Okay, thanks, Gus. So the group I'm now trying to set up, and so I've just started setting up my group in Oxford, and then with a lot of activity in Uganda and Kenya. The overall sort of theme of the questions that we're trying to answer is how we can use human genetic tools to better understand the causes and the consequences of childhood infection. So just to give that a bit of background, so as you'll appreciate, infection causes over 3 million childhood deaths annually, and the bulk of those deaths occur in children living in low and middle income settings, in particular in sub-Saharan Africa. So we urgently need better tools to control infection, particularly in low income settings. But alongside those infections from which that result in mortality, there's inordinately more infections that children survive, and we've really got a very poor understanding of what it means for a child growing up with a high burden of infection for their long-term health. So I think to achieve better child health outcomes with respect to infection, we need to understand why it is some children are at risk of severe and invasive infection, but we also need to understand what the long-term consequences of that are. So I'm going to try and illustrate those ideas with work that I've been doing for about the last 4 or 5 years, thinking about those sorts of themes with respect to rotavirus disease and immunity. And so rotavirus, as I suspect you all know, is the most common cause of acute watery diarrhoea in the under-2s. It continues to have high associated mortality, in particular in sub-Saharan Africa. And those numbers are from about 10 years ago, but they remain the same today. And that's despite having available and deployed rotavirus vaccines. And the reason for that ongoing mortality is that while those rotavirus vaccines work very well in high-income settings, in low-income settings, they've got very poor efficacy. And the reasons for that disparate efficacy are poorly understood and is very likely to be multifactorial. But over the last decade or so, there's been increasing interest in whether human genetics can help explain some of that or tell us something about that. And the reason for thinking that that might be the case really comes from some molecular biology that was observed about 2 decades ago, which is that the majority of rotavirus strains infecting humans, when they do so, rotavirus attaches, invades and causes disease depending on its P antigen by binding human blood group antigen expressed on mucosal surfaces. And the reason then for being interested in human genetics with respect to that is that in human populations, about a quarter of individuals won't express human blood group antigen on the mucosal surfaces, so they're termed non-secretors. And so from that, you'd predict that those individuals are almost entirely protected against rotavirus infection and disease. And those predictions have been borne out by a series of candidate gene studies over the last decade or so, and the results of those have been really very consistent. So on the left-hand side here, for P8 genotype rotavirus disease, this is coded with respect to being a secretor. If you're a secretor, so if you put human blood group antigen on your mucosal surfaces, compared to children that don't, you're at enormously increased risk of rotavirus disease for P8 genotype disease, and that's both in vaccinated populations and unvaccinated populations. For P6 genotype disease in the middle, that makes absolutely no difference. So P6 rotavirus doesn't need human blood group antigen to mediate invasion. And then on the far right-hand side, because rotavirus vaccines are oral live attenuated vaccines and they're monovalent PA genotype vaccines in the large part, for you to have a vaccine response to Rotarix, for instance, you need to express human blood group antigen on your mucosal surfaces in order to generate that vaccine take, which is what that's demonstrating on the right. And just at this stage, it's worth just reflecting a little bit on what I think is a slightly counterintuitive relationship between genetics and vaccine take and disease risk here. So if you think about this in the other way round, so non-secretors don't express human blood group antigen on their mucosal surfaces, and so they've got very poor vaccine responses. But actually, it doesn't matter for that subset of children because they're at no risk of disease in the general population. So interpreting what vaccine take means for infection risk without genetics in this context is almost impossible. Okay, so that's been really helpful in terms of defining the role for secretor status in terms of rotavirus infection and immunity. But it doesn't tell you whether there's a broader role for human genetics in those traits. And to sort of try and better flesh that out, we performed a series of genome-wide association studies for rotavirus disease and immunity across 3 settings about 3 or 4 years ago now. So the first of those genome-wide association studies was in Uganda within Alison Elliott's Entebbe Mother and Baby study. And the EMAB study started off as a trial of about 2,000 pregnant women in an RCT of anti-helminth treatment. And then those infants were then followed through early childhood with a particular focus on infection phenotypes. But what it sort of blossomed into is this really incredible birth cohort with fantastic regular sampling throughout childhood and really detailed phenotyping throughout childhood. And these guys are now sort of in their sort of early 20s. And so using that, what we were then able to do was take samples from children at ages 2 and 3 and quantify rotavirus serum IgA, and then pair that with genome-wide genotypes to do a genome-wide association study of what is naturally acquired immunity. So this is all pre-rotavirus vaccine rollout. We could then do the same thing in a Zambian population, so slightly smaller number of children, but exactly the same idea. So Zambian children that have been recruited to a Rotarix trial of 2 doses versus 3 doses, and we've again assayed anti-rotavirus IgA at the age of a year. And then we can do a genome-wide association study of that. So trying to get at what the human genetics of vaccine-derived immunity looks like, or at least immunity in a vaccinated population. And then finally, we wanted to understand whether what the genetics of hospitalised disease looked like. So we did a third genome-wide association study of hospitalised rotavirus disease in coastal Kenya, about 500 hospitalised cases and about 1,500 population controls. And I appreciate that most of you will have a good understanding of what a genome-wide association study is, but just to make sure that we're all on the same page, when you're doing a genome-wide association study, all you're doing is you're typing common genetic variation in all of your cases and controls, or all of your samples included in your study. So you're typing at regular intervals right the way across the genome, millions of single base pair changes, and then all you're doing is you're asking whether carriage of one allele or another, each of those base pair changes is associated with whatever outcome you're interested in. So it's just a really simple regression analysis that you just recapitulating millions of times across the genome. And what you get out of that then is this. So this is a Manhattan plot, and this is our Manhattan plot for anti-rotavirus IgA in Ugandan children. So each dot represents an association test at a SNP. How high it is on the y-axis is the negative log to the base 10 p-value, so how associated it is with anti-rotavirus IgA. And what you're hoping to see are peaks that come above this red line, so genome-wide significance. So you've got a significant peak on chromosome 19 and a significant peak on chromosome 2. So that chromosome 19 peak represents secretor status, as you kind of expect. And I don't think that, it confirms that that's important in Ugandan children, but, doesn't really advance our understanding of the biology of rotavirus infection particularly, although it's nice to see as a positive control. And the lead SNP for that is the lead SNP that you'd expect. So it's RS601338, which is the major determinant of secretor status in most human populations. So that introduces a G to A change. It's a nonsense mutation. So you get a truncated copy of FOOT2.
I don't entirely know what to do.
No, I don't think I've trodden anything.
No.
Oh, I'm still there.
Huh. Oh no, it was working, that's my fault. Yeah.
Cool. Okay.
I did.
Like you can.
Yeah.
Well done. Yes, and what you can see is that when you stratify rotavirus anti-rotavirus IgA responses out by genotype at secretor status, the relationship's kind of what you'd expect it to be. So it doesn't really matter if you've got one functional copy or two functional copies. What matters is if you don't have any functional copies. So it's very much a recessive effect. So it's the AA homozygotes who are the non-secretors, and everyone else behaves like a homogeneous group, regardless of whether you're a major homozygote or a het. What's perhaps more interesting, though, then, is that we had this second locus on chromosome 2 that hadn't been previously described as being associated with rotavirus disease. And so it's a C to T change. It sits in an intron in a gene called EDAR. And children in the general population who are carrying a T allele at this locus have better, on average, immune responses at the age of 2 and 3 in Uganda to rotavirus. So this is naturally acquired immunity, so none of these children have been vaccinated. Novel locus, so you want to try and replicate that in some way, and we can replicate that, albeit in a far smaller number of children in Zambia. So in Zambian children who are vaccinated at the age of 1, again, that T allele is associated with better immune or greater immune responses to rotavirus. Okay, so having identified and validated a novel variant associated with rotavirus immunity, there's several ways that you can think of to then move that forward to tell you something interesting about the disease biology. One of those would be to sort of pair your GWAS data with expression quantitative trait locus mapping and try and understand what the gene is that's causal of that and try and leverage that in some way to tell you some new biology. And we've obviously done a bit of that. But what I'm going to focus on is how you can use that knowledge that you've got a group of individuals who, on average, make better immune responses in the population you can identify through their genetics to think about whether you can link then immunity to disease risk and then beyond that to sort of indirect effects of rotavirus vaccine control. So by having this group of individuals in the population who are these T allele carriers who have better immune responses, the first thing you can then do is ask whether, in a Mendelian randomization framework, whether those same children are then protected against disease, to give you some causal evidence that making better immune responses protects you against getting hospitalized rotavirus disease. And obviously we can do that within the Kenyan data, because we've got a genome-wide association study with hospitalised rotavirus disease. And so when you ask then whether those T allele carriers in Kenya are protected against hospitalised rotavirus disease, they are, albeit only for P8 genotype rotavirus disease. And it's worth, again, thinking about the direction of effect here, because it's distinct from what you see in secretor status. So children that are making better immune responses here are the T allele carriers, and it's those children that are protected against disease, which makes a great deal of sense, I think. These are all unvaccinated children, both in Kenya and in Uganda. So if you're genetically predisposed to make better immune responses, and that's going to protect you against disease, what you would predict is that is going to accrue with your exposures to rotavirus. That won't happen for your first exposure. That's going to happen for your second, third, fourth exposure. Obviously, we don't know what the personal rotavirus history are of all of these children, but obviously we do have their ages. So as a proxy of trying to sort of think through whether this is likely to be primary protection or secondary protection, we can then stratify out into younger infants and older infants and ask when that protection is turning up in the population. And you kind of see what you'd expect to see. So that protective effect of making better immune responses accrues with age. So you see no protective effect in children that are under 12 months in Kenya, but as they move into their second year of life, you start to see a protective effect of making better immune responses.
James, can I ask a really stupid question?
Yes.
How much does serum IgA correlate with gut IgA?
No, it's not. It's a really good question, and actually, your talk at ESPID actually made me go away and read some papers about that. And there's really quite old papers around that, and it doesn't correlate desperately well with fetal IgA. It does actually correlate better than it seems to me from what you were saying for respiratory pathogens with salivary IgA. So anti-rotavirus IgA in serum is a bit problematic. Yeah. It works quite well in places where rotavirus vaccination is working as a correlative protection. Outside of those settings, it really doesn't work very well, where I think there is some of the utility of this lies, actually. It's trying to pick that apart a bit. So we can demonstrate that then that you can link better immune responses to rotavirus to disease risk. And what we're also able to then do within EMABS is, because we've got all of this really quite detailed, often infection-related phenotyping that's been done on an annual basis in terms of questionnaires for the EMABS participants, we don't have rotavirus-specific diarrhea data, but we've got all-cause diarrheal disease data. So what this is showing you on the right-hand side here is the frequency of diarrheal disease episodes, well, actually the total number of diarrheal disease episodes from the age of 0 all the way through to the age of 10. And that's stratified out by genotype of this locus. And I think, unsurprisingly, you don't see anything. And the reason I don't think that's desperately surprising is if this is a rotavirus-specific effect, there's lots of causes, especially through to the age of 10, for diarrhea that is unlikely to be modified by immunity to rotavirus. But if you stratify that out by subtypes of diarrhea, what you see is this protective effect against diarrhea with dehydration, no effect for diarrhea with dysentery, no effect for prolonged diarrhea, which of course is what you'd expect for a rotavirus-specific effect. Of course, it causes acute watery diarrhea, doesn't cause dysentery, doesn't cause prolonged diarrhea. Okay. So linking, I think, immunity to disease through genetics is a useful thing to be doing, but I think it doesn't tell you something that you weren't expecting to see. What I think is more interesting to ask, using the same framework, is whether we can link rotavirus using genetics to other health outcomes in the population. And the way that I've chosen to do that to start with, and I'll come on to the pitfalls around this later, but the way that I've chosen to do that to start with is to go back to using secretor status as an instrument for that. And the reason for that is that we know lots and lots of populations that secretor status is critically important as a determinant of rotavirus infection and disease, and the effect size is huge. And you can see that in terms of serology in Uganda, which is the data here I've shown you before, and you can see that in terms of Kenyan children for hospitalised disease. So Kenyan children who are non-secretors are almost entirely protected against hospitalisation with rotavirus disease. So it's a really strong instrument. And the advantage of working with researchers in Kilifi is there's lots of really strong infection genetics data that's already there, and infection epidemiology and infection surveillance data that's already in place. So what we're able to do then is to use all of that data to then ask whether protection against rotavirus disease is also protecting you against other things, specifically here protecting you against bacteremia. And so we're able to do that because we've got GWAS data from 1,500 children with community-acquired bacteremia and about 2,500 population controls. And that's set up, so that obviously in order to get 1,500 children with bacteremia, that's over a long period of time. So all of that collection activity set against this period of really quite profound changing infection epidemiology in Kilifi. So you've got this huge decline in malaria, You've got bacterial conjugate vaccines turning up. You've got rotavirus vaccine turning up in 2014. And also really detailed surveillance for lots of viral infections, both respiratory and enteric viral infections right the way across that period. So we know in reasonable detail what's happening in the community in terms of infection transmission. And laying that on top of the genetic data then allows you, I think, to make sort of stronger causal inferences, which I'll come on to. So, if you ask whether being a non-secretor is protecting you against bacteremia, overall it's not. What it specifically seems to do is protect you against gram-negative bacteremia. And if just for a moment you accept that this is being mediated by rotavirus disease, that makes a great deal of sense. Rotavirus causes intestinal inflammation, it's going to increase gut translocation, and that's what you would expect to then see. Gram-negatives are gut organisms, or commonly gut organisms, and so an association with gram-negative bacteremia makes a reasonable amount of sense. The problem, of course, though, is that what you don't know is whether there's some direct effect of being a non-secretor on enteric bacteremia risk, because we know that, for instance, secretor status also affects your gut microbiome. So you can imagine a scenario in which your likelihood of being colonised with some enteropathogen or other is more likely whether you're a secretor or a non-secretor, regardless of your risk of rotavirus disease. But what we can, and this is where the infection surveillance within Caliphie then really comes to the fore, because what you're able to then do is to then overlay that analysis on top of rotavirus surveillance in the community. So much like when we had lots of rotavirus in the UK, there's a strong sort of seasonal pattern to rotavirus transmission in Caliphie. And this is only across 3 years of the data that we've got here. You've got strong seasonal variation. So what you can then do is you can ask whether that protective effect of being a non-secretor on bacteremia risk depends on the transmission of rotavirus in the community. You can do that by stratifying out your analysis. So you can ask whether you see that effect in low transmission periods or high transmission periods, and you only see that protective effect of being a non-secretor when rotavirus is circulating at high levels in the community, suggesting that rotavirus is a determinant of gram-negative bacteremia. And if you can better control rotavirus, you'll better control enteric bloodstream infection. Okay. But I said at the start that what I'm really interested in trying to do as I set up my fellowship is thinking about what childhood infection means for long-term health, because all of what I've told you about is all very proximate. It's all happening at around the same time, probably within the same couple of weeks. What I think is more challenging, but potentially more interesting, is to think about what a burden of childhood infection means for how you handle or how non-communicable disease risk is expressed in adulthood. And on the face of it, secretor status seems like a good place to start with that. So if you wanted to understand what risk of rotavirus disease and diarrhea does for long-term health outcomes, we know across lots of populations that being a non-secretor protects you against rotavirus disease. So everything coded here is coded with respect to being a non-secretor. So you're protected. So serological data is in orange here, and then infection data is in green. So you're protected against rotavirus infection and disease if you're a non-secretor. In Uganda, in terms of all-cause diarrheal disease, you're very much where you're protected significantly in the first year of life, and then that wanes beyond the first year of life. And similarly, in European ancestry populations, you got very strong protection in that first year of life that then starts to wane in the second year of life. The trouble, though, with using secretor status to try and infer something about rotavirus infection and long-term health outcomes is there's enormous pleiotropy at that locus that has nothing to do with, at least I don't think it has anything to do with rotavirus. So we know that it's a huge determinant of rotavirus infection, but it's also a very significant determinant for lots of other viral infections. So again, serological data is in orange here and infection data is in green. And so if you're a non-secretor, as well as being protected against diarrheal disease, you're at least from a serological perspective, at greater risk of polyomavirus infection, so JC virus and BK virus infection, also HHV6 infection. You're also at, so this is mostly questionnaire-based data down at the bottom, you're at increased risk of measles and mumps. And I think that's interesting in terms of thinking about what's happened evolutionarily here, because the secreted state of polymorphisms sit at about 50% allele frequencies in almost every population, and I think the reason for that, is if you've just got selection that's being imposed by protecting you against diarrhea in infancy with high mortality, that's going to move to fixation pretty quickly in population. So you're going to have some sort of balancing selection. I think measles is a pretty good candidate for that.
I'm amazed we got enough measles in Elsbach. 9,000 kids born in 1990 in Bristol. No, it's just incredible. Yeah.
Oh, and the effect estimate's really consistent.
Oh, vaccination, the measles vaccine of 17, I think. I don't know, you guys would know better than me, but I just didn't think we had that many cases. Maybe we did.
You've never been around Montpellier.
No, I live in Montpellier.
Don't want to get vaccinated.
Yeah.
The school was slagged off vaccination. Anyway.
So I think it's I think secreted status can't implicate a single infection in long-term health. But what I think it can do is tell you that how your infection history is important for your long-term non-communicable disease risk. So there are big effects of being a non-secretor on your long-term non-communicable disease risk. And by the time you're an adult, being a non-secretor is pretty uniformly bad for you, so at least at the population level. So you're at increased risk of type 1 diabetes, Crohn's disease, peptic ulcer disease, coronary artery disease, and that cardiovascular risk is sort of borne out by increases in a whole heap of cardiovascular risk factors, so cholesterol, baseline CRP, blood pressure. So I wanted to sort of deviate a little bit at this point and just explain the experiment that got me really interested in the idea that how your personal infection history might set up your long-term non-communicable disease risk. And this was an experiment I did with Ben Fairfax a couple of years ago in Oxford. And what Ben had done was he'd taken close to 200 individuals, healthy individuals, in sort of somewhere between about they're sort of in mid-adulthood, sort of median age of about 40. Isolated monocytes and then cultured those monocytes either in the presence or absence of innate immune stimuli. So we started off with a panel of sort of LPS, PAM3CSK4 and interferon. And what we were after with this experiment, we were trying to define in a reasonably large population, which hadn't been done before at this stage, what the epigenetic consequences of innate immune stimulation for monocytes were. And so we measured that by assaying DNA methylation in paired samples, so untreated monocytes and monocytes stimulated with something. So what we got out of that was, so this is the pilot experiment, so this isn't the full 190 individuals, this is just in 30 individuals. And even just within those 30 individuals, so what you're seeing at the top are principal component plots of CpG, methylation, genome-wide, coloured by what you've done to the monocytes. So untreated is in green. And you can see even at sort of 30, you're sort of starting to pull apart those different stimuli. But when you look at sort of the individual CpGs that are significantly methylated, hypermethylated or demethylated, actually that's kind of dominated by LPS. So actually most PAM3CSK4 differentially methylated CpGs are captured by LPS, which from a pragmatic and cost perspective meant that what we went on to do for the whole cohort was just look at LPS. So what I'm showing you from now on is just with and without LPS treatment. And what you see when you, across a population of a couple of hundred individuals, when you stimulate their monocytes with LPS and then assay DNA methylation, a huge epigenetic changes right the way across the genome. And they're dominated by demethylation events. So about 2/3 of those events are demethylation, and where those demethylation events in particular sit are in enhancers. And the same is true, although the enrichment's less significant, of hypermethylation events. So what you sort of would predict that then doing is for those demethylation in enhancers is permissive of local gene expression. So it's turning on stuff in response to LPS. And then similarly, hypermethylation should be repressive. And in keeping with that, if you ask then what the local genes are to those demethylated CPGs, the things that you're turning on, the things that they're enriched for are very much what you'd expect. So TLR signalling pathways, innate immune signalling pathways, cytokine response pathways. But what at least I think is really neat is that for methylated CPGs, you see absolutely no enrichment for anything at all, which I think makes a great deal of sense. So the things you turn on are going to be specific, but the pathways that you need to turn off when you're responding to LPS are quite diverse. So there's not a single pathway correlate that gets enriched. It's turning down lots and lots of different things. What was more unexpected, though, was those genes that are local to demethylated CPGs are hugely enriched for cancer-associated genes across a range of cancers. And gives you is this hypothesis that then perhaps episodic inflammation through your life, perhaps triggered by infection, could be leading to epigenetic changes in your immune cells. And then is that then impacting on your cancer risk in later life? And clearly that doesn't show that, but it gives you a sense that that might be something that's worth exploring. Alongside that, there are many things you can do with methylation data, but another thing you can do with methylation data is you can calculate epigenetic age. So the idea that there are lots of different epigenetic clocks, but the oldest one is this one. So this is the Horvath clock. It uses about 300 CPGs, and it gives you remarkably stable estimates of age, almost regardless of what tissue you do it in. When you get your age estimate in a population, that correlates really very precisely with chronological age, which on its own doesn't sound like a desperately useful thing to be doing. But where they are really interesting is that your epigenetic age, if it's accelerated as compared to your chronological age, so you've got epigenetic age acceleration, that's enormously predictive of a wide range of bad outcomes. So early mortality, cardiovascular disease, cancer, dementia, lots and lots of things. So we're interested in measuring what LPS was doing to epigenetic age estimates, and what we saw was you've got lots of demethylation events, so perhaps this is not that surprising, but LPS treatment ex vivo induces epigenetic age acceleration by about 6 months. And you can imagine perhaps that might just be a non-specific effect. You're perturbing a lot of CPGs. But what got us really interested in this was actually you don't see it in a generalised way right the way across the cohort. The individuals that respond to LPS and accelerate their epigenetic age are individuals for which that hasn't already happened. So if you've already got epigenetic age acceleration, LPS doesn't do anything for you. It's only those individuals who are yet to have that epigenetic age acceleration, suggesting then what you're doing ex vivo there is perhaps phenocopying something that's already happened for your epigenetically accelerated individuals in their earlier life, perhaps being infection burden. And that's what then sort of brings me back to secretor status, because as I said earlier, secretor status isn't, I don't think, very tractable as a tool to link a specific infection to a long-term health outcome. But what, of course, it does do is it gives you two groups, it allows you to divide up two groups of individuals, secretors and non-secretors, who will have had profoundly different infection histories. You can't tie that to a single pathogen, but their infection histories on average will be different. So you can then go back to our methylation data and ask then whether that epigenetic age acceleration then induced by LPS is confined to secretors or non-secretors. And that's exactly what you see. So Non-secretors, if you treat their monocytes with LPS, you induce no epigenetic age acceleration, and that epigenetic age acceleration is only seen in secretors. And the way that that appears to be working is you've already accrued that epigenetic age acceleration among non-secretors. So what I was then interested in thinking about was, okay, so that's only taking in essence 300 CpG. So is this a generalised effect on baseline methylation of being a secretor or a non-secretor? And in terms of the average baseline CpG methylation, you see no effect of being a secretor or a non-secretor genome-wide. So the middle bit here, what I've done is overplotted the median CpG methylation state at each of 400,000 CpGs. And so this is the sort of bimodal distribution that you get. And there are two lines there, you just can't see the blue one behind it because they perfectly overplot. So there's really no effect in terms of the medians. In terms of individual CpGs, the only thing that you seem to robustly see is an effect for secretor status on CpGs that sit in the FUT2 promoter. So that makes a great deal of sense, but nothing generalised across the genome apart from one other CpG. What's interesting though is that that's what you see in terms of average CpG methylation. But what it does seem to, or what secretor status does seem to perturb, is baseline CpG methylation variance. And non-secretors, that's shifted so the variance is lower, which is what I'm showing you on average in the middle. And then in terms of individual CpGs, you see not a huge number, but a handful that are differentially, that have differential variance at baseline according to secretor status. And again, you pick out some things that almost certainly have nothing to do with infection history. So again, FUT2 promoter and also the ABO promoter, which you kind of expect biologically, but a reasonable number of other things. And thinking about CpG methylation variance seems like a pretty esoteric thing to be thinking about. But the reason that that we wanted to look at this was from the original methylation experiment. What we'd seen was that the major determinant of whether or not a CpG responds to stimulation is its baseline state, and it's not its average baseline state; it's its variance. So here on the left-hand side, what I'm showing you is the average variance. It's the baseline variance of CpGs grouped into at the bottom, those that don't respond in grey, and increasing number of conditions they respond to as you move up that. So the more inflammatory stimuli that a CpG responds to, the higher its baseline variance. Okay, as I said earlier, DNA methylation is enormously predictive and changes transcription. So I guess then the obvious question is secretor status perturbing transcription in immune cells? And we can test this to a degree, again, using data that was generated through Ben's lab, with a very, very similar experimental setup. So 190 odd individuals, isolate monocytes, culture those monocytes for 24 hours with and without interferon gamma. And then we've assayed transcriptomes in all of those monocytes with RNA-seq and got paired genome-wide genotyping data from which we can infer secretor status. So at baseline, secretor status doesn't appear to modify monocyte transcription. So this, what this Manhattan plot is showing you is that each dot is a gene and how associated it is with secretor status, and there's no significant association. So obviously your transcriptome in your monocytes, or indeed any immune cell, is highly context dependent. So when you stimulate those monocytes with interferon, almost 80% of the transcriptome is perturbed by stimulation with interferon. Not, again, a huge surprise. But what is interesting is about 100 of those genes, their upregulation or downregulation in response to interferon significantly interacts with secretor status. And this is the sort of thing that those genes look like. So you have an interferon upregulated gene in secretors that's highly significantly upregulated, but you see no difference in non-secretors at all. And what that seems to mostly be driven by is that upregulation that you're seeing in secretors has already happened at baseline in non-secretors. So what I think that gives you then is this hypothesis where your infection history, proxied here by secretor status, is subtly perturbing immune cell function through perturbations of baseline epigenetics and then reprogramming of transcriptional responses to inflammation. And that's kind of everything I wanted to take you through. So I've told you just very briefly about a couple of different projects that I think are sort of related in some ways to each other. So firstly, I've shown you how we've identified novel genetic variation associated with rotavirus disease and immunity, but also confirmed a role for secretor status in Ugandan, Kenyan and Zambian populations. But what's been, I think, far more informative is that we've been able to use that to link immunity to disease and also understand the wider indirect health benefits of potential improved rotavirus control, in particular with respect to gram-negative bacteremia. And then secondly, what I've taken you through is how innate immune stimulation induces profound epigenetic and transcriptional changes in immune cells, but I think those are being modified by an individual's infection history. And then I think what that gives you then as an overarching model is what I've sort of set out on the left-hand side there. So I think a child's germline genetics determines whether or not they are likely to develop severe and invasive infection, but in turn their infection history through childhood is shaping their genetics through epigenetic changes, in particular in their immune cells. And I suspect that's very likely to have consequences for their long-term non-communicable disease risk. And those are ideas that I'm hoping to sort of flesh out during my fellowship over the next 6 years in Oxford, and also with lots of activity in Kenya and Uganda. And really what we're trying to do with that is to expand those ideas out to a broader set of infections among children with a high burden of childhood infection. Obviously, putting all of that together takes a lot of people, and I'm really lucky to work with lots of fantastic collaborators in Uganda, in Kenya, in Zambia, and also in Oxford. Thank you.
Thanks, that was super interesting. That was great. Have we got some questions? Any questions from the floor? All right,
Thanks, James. Really great talk. Really fantastic progress you've made. What's EDAR?
Oh, yeah. Yeah. So it's interesting. So it's one of the causes of ectodermal dysplasia. It has nothing to do with anything, actually, I don't think. So it's, although it's a really nice example of what your regulatory variance is local to actually isn't necessarily what it's doing.
Gotcha.
So when you pair that with EQTL data, actually what you see is that it's a significant EQTL in monocytes for LIMS1, which is about 200 kilobases downstream, which is a sort of integrin adapter protein. I'm not entirely sure what that's doing, and I'm still sort of thinking.
I noticed I think between those two you had RAN-BP2.
Yeah.
Which is also an interesting gene.
Yes,
it's a nucleophore gene, isn't it?
Yeah, absolutely.
I don't know. Do you think it influences expression of that?
No, I don't think so, or at least I mean. So a huge amount of, so a lot of what I'm going to try and do with my fellowship is thinking about what I sort of set out there, but a huge amount of where I think there's a hole in terms of genetics in African populations. It's not actually around genome-wide association studies. It's about EQTL studies and functional genomics to understand that. Because at the moment, you're really just stuck with European ancestry stuff to try and annotate things. And so what is there at the moment? No, I can't see anything. But the fact is, it's relatively rare in European populations. So you're asking a lot of the data sets, even assuming that the regulatory environment is the same, but even assuming that, you're asking a lot of data sets because in European populations, it sits at just under 1%. It's about 15% in African populations. So yeah, I just don't think we've got the data.
Was that EDAR you were talking about?
Yeah.
I thought that was like, I just thought that was a classic hair thickness,
Yeah.
tooth eruption.
Yeah.
to select a
Yes, exactly. And so so loss of function variants cause ectodermal dysplasia.
Yeah.
I got very excited about it when I saw it, and I thought it was likely to be EDAR because it's one of the along with lactase persistence, it's one of the examples of really, really profound selection pressure. So there's a huge selection pressure at that locus. But the most of that evidence for that selection pressure is for allele frequency differentiation of exonic mutations in EDAR. So I think if this isn't mediated by EDAR, I think that's oddly irrelevant. But yeah, I spent about 6 months thinking that was the case.
Have they actually got ancient DNA data to actually look at allele frequency change, over thousands of years, or is that genomic evidence of something?
That's just genomic evidence across. Yeah, exactly. So yes, it may be that actually the allele frequency differentiation at those exonic SNPs is actually marking something else. I think that's very, very possible.
But the bottom line is all rapid selection, all of it is due to infection, including lactose persistence.
Yes.
It's all due to infection.
Yes, and probably diarrheal disease.
Any further questions online?
I have a question. Okay. How specific is your relationship between epigenetic age acceleration and secretor status to the Horvath plot?
Yes, no, great. I've not done it exhaustively, but of the ones that I sort of have got set up, of which there are about 6, you see exactly the same thing. The reason I've used the Horvath clock is that's what we published on originally for the methylation paper, and it's one of the best established. But no, you do see it across sort of, what do we do, sort of grim age and pheno age and those sorts of things. You see the same. Obviously, your estimate of how much you're accelerating in terms of months or years is different, but you see the same pattern of effect.
And have you looked at other cell types as well?
No. And I think that would be really, really interesting to do.
Just about the measles and Alzheimer's mystery, that's because that's the Alzheimer's mothers, not the Alzheimer's kids.
Yes.
But I think in terms of any sort of infection genetics, including all these claims from 23andMe, etc., it's importantly illustrative because the Alzheimer's mums hardly any will have had measles vaccination. So that means that 98% of them would have been infected by the age of 15 with measles unequivocally. Yeah. And 100% by the time they were a kid.
Yeah.
Therefore, you're not looking at the fact of infection at all. In those studies, you're looking at people at reporting having been infected, which will relate to how much the kids actually were bothered about being ill. Yeah. Probably something it'll be saying something about severity, but it's not telling you about infection. Yes, no. Even though these get reported as being infection, yes, no, it's not. It's everyone would have got every single person got measles. Yes. So it's. But that means what you're looking at is the genetic. So then you might expect to get some sort of genetic correlations with things like just general reporting tendency. Be interesting to look at that in relation to kids reporting being unhappy as kids or whatever. Because you've got these are sort of people in their 20s and 30s reporting back on whether they remember actually remember being in bed and feeling unhappy.
Or perhaps like a determinant of like age of infection or something like that. You're more likely to remember something that happened to you when you're 7 or
that's interesting. The mumps thing would definitely be that.
It's quite
yes, because I very clearly remember having mumps at the age of 8 because
You didn't get MMR?
Well, I'm older than you.
It's really interesting epigenetic results. I was wondering about, so cell lines or cell culturing has really profound epigenetic effects. Is there any chance, or are you planning to do any of those kind of looking at those effects outside of cell culturing?
Yes, all of those stimulation effects on methylation, they're all done in paired. So I completely accept that doesn't entirely obviate cell cut, but they've been treated in the same way, just with or without the stimulus, and it's all the paired analysis. And certainly when you sort of try and model whether you've got any, what your false discovery looks like, as you'd sort of do with any EWOS or whatever, I don't think we've got too much inflation from technical stuff. But yes, absolutely. And I think, of course, what I'm showing there is what's happening ex vivo. I think what's critical is trying to understand what episodes of infection do actually in terms of wild type infection. So something that obviously so the OVG is really strong at is human challenge models. So one of the things that I'm trying to leverage locally is just to ask whether you can see infection-induced methylation changes in immune cells before and after controlled infection. That obviously has some limitations because it's very controlled infection. These are not sick people, but I think that's, it's a reasonable way to start.
Any more questions from the floor or online? So I'd make sure I haven't forgotten you online. Because if not, I've got a question.
There you go.
I was just wondering how I'm just going to check. There's nothing happening online.
I think Lindsay's watching.
Oh no, it's just a recording. Okay, great. How obviously non-secretor or secretor status is a very nice instrumental variable that you reflect some of its possible weaknesses. Are there likely to be many more of these? How unique do you think that secretor status is? Lactase resistance is one other potential plausible variable. Do you think that we just simply haven't done enough GWAS in settings where there's huge amounts of infection burden and we're going to pick up, or we start with what we got?
I think whenever we've done it properly at a pathogen-specific level, I think you found stuff. So sickle cell trait, is a fantastic instrument in terms of thinking through malaria, and I think actually really when you look at what we've done in terms of pathogen-specific GWAS, I think it's very, very little. I think it's underpowered. I think when you get up to reasonable sample sizes in well-characterised cohorts, and I think that's key. I think trying to do this without having a really good understanding. So one of the things I'm trying to do at the moment is to see whether we could get an instrument for RSV disease. And I think one of the real weaknesses of a lot of the data that's tried to do that before is that you don't have a good handle on size of baby, age of baby, really critical determinants of whether or not you get RSV infection. And it may be that there isn't anything at all. Maybe those are the determinants, but I just don't think we've looked.
Okay, interesting. Thanks. Any other questions?
I'm just wondering if you probably thought about this, but can you use the epigenetic age or the methylation readout to try to get a cumulative measure of exposure, or is the sort of limit of the value giving you time? You won't be able to recapitulate time of infection, but obviously you could then go back and look at historical samples. I'd be interested to sort of think.
So yeah, I think I guess the way that I'm thinking about that, I guess will change over time. But some of the things that I'm trying to look at the moment in terms of thinking that through are making use of that EMABS cohort, and because you've got really nice infection data and stored samples, trying to correlate methylation trajectories over early life and understand that. And then also within another of Alison Elliott's trials, look at, for instance, whether malaria chemoprophylaxis for a year does anything in terms of methylation, just to try and sort of tie that down in sort of different ways. So yeah, very much.
What prophylaxis are you using? Do they have methylation change itself?
I can't remember.
Yeah, all right. Any other questions? If not, I think we can probably just thank James for coming. Appreciate you coming. I think he's here all afternoon, probably quizzing in more detail by malaria prophylaxis. Yeah. Thanks. Cheers.
Cheers.
Yeah, great. Super interesting.
Cool.
Yeah, really nice. Yeah. I'll leave it.